We spoke about two general divisions. Okay. We talked about the autonomic division of the peripheral nervous system, and we talked about the somatic. Okay. And I'm going to draw a couple of neurons here. Okay. Now, what were the subdivisions of the autonomic nervous system? What were the two divisions of the autonomic nervous system? Parasympathetic and sympathetic. Okay. And we said that in the parasympathetic and sympathetic system, there were two neurons that were connected. Okay. There was a preganglionic neuron for both of these, and a postganglionic neuron. Okay. In the somatic nervous system, what was the neuron? A motor neuron, perfect, a motor neuron. Okay. Now, what was the effector tissue of the autonomic nervous system? What type of tissues did the autonomic nervous system have effect on? All right, so smooth muscle. What else? Cardiac muscle and adipose, right? And glands, yeah, glands. Smooth muscle, cardiac muscle, glands for these parasympathetic. Adipose was only for the sympathetic, okay? For the somatic nervous system, what was the effector organ or tissue? Skeletal muscle, perfect, skeletal muscle, okay? We said that this was voluntary or involuntary? Involuntary, so it was not under our voluntary control. And the somatic was voluntary, so we could control the somatic, the skeletal muscle movements. Okay. Now, let's talk about the spinal cord segments here. So we're in the, let's try to draw this out. I'm going to draw this really small here. Okay. Now, where in the spinal cord did the fibers of the autonomic nervous system arise from? The lateral horn of the spinal cord. Thank you. So I'm just going to draw an X here. All right, so the fibers came from the lateral horn. What about the somatic nervous system? The ventral horn. Perfect. So I'll just draw an X here. All right, so the lateral horn versus the ventral horn. Now, what area of the spinal cord did the sympathetic fibers come from? What levels or what segments? Thoracolumbar. So that was T1 to, that was T1 to L2, right? What about the parasympathetic system? So we had some cranial nerves there. What cranial nerves? 3, 7. Cranial nerves 3, 7, 9, and 10. And we had pelvic nerves. What pelvic nerves? S2, S3, S4. Okay. All right. Now let's talk about some of the communication here. So we said in the sympathetic nervous system, right? These two neurons communicated at what? What was this structure called? Generally, it was called an autonomic ganglia. Right, so this slide here, autonomic ganglia. Now, what was the specific name of the connection or collection of autonomic ganglia in the sympathetic system? What was that structure called? The sympathetic chain, okay? So this is the sympathetic chain. Okay, the sympathetic chain. And that was only one route of communication. What were the other 3 routes of communication for the sympathetic preganglionic neuron? So 1 is it could synapse on the sympathetic chain. 2, a collateral ganglia, right? A collateral ganglia. Okay, and that's gonna go to your GI organs. 3, what was the third option? The adrenal medulla. The adrenal medulla. Right, the adrenal medulla. And that was going to release norepinephrine, about 20%, epinephrine, about 80%, and those are going to be hormones. Okay. All right. Now the parasympathetic only had one route of communication, right? Now let's also look at the length of the neurons here. We saw that in the sympathetic system, because the sympathetic chain was so close to the spinal cord, they had shorter preganglionic neurons and longer postganglionic neurons. And the reverse was true for the parasympathetic system. These longer preganglionic neurons went all the way out to a ganglia that was close to the effector organ, and then a very short postganglionic neuron went to that effector organ, okay? Now, let's talk about the receptors and the neurotransmitters here. What was the neurotransmitter that we said both preganglionic neurons released? Acetylcholine, right? Acetylcholine. Good. And it was going to bind to a receptor on the postganglionic neuron. What type of receptor was that? What type of receptor was that? So because acetylcholine is going to be cholinergic, but what subtype of cholinergic? Nicotinic, right? Nicotinic. Just write that here. Okay, and what that means, these nicotinic receptors, they are also acting as channels, okay? So when acetylcholine binds to these nicotinic receptors, in a very rapid way, that channel will open, sodium will rush in, potassium rushes out. That will immediately depolarize the second neuron, and another action potential is going to be fired, okay? Now, let's look at the neurotransmitter released from these second neurons, the postganglionic neurons. What was the neurotransmitter here? What was the neurotransmitter of the sympathetic postganglionic neuron? Norepinephrine. Oops. Norepinephrine here. And what about the parasympathetic postganglionic neurons? Also acetylcholine. Also acetylcholine. But what type of receptors were on the effector tissue in the parasympathetic side? I'm sorry, sympathetic side. What was adrenergic receptors? Right? These were called adrenergic receptors. And what were the receptors on the parasympathetic side? Muscarinic, cholinergic. So they were also cholinergic because they're going to bind acetylcholine, but they were a different type of cholinergic. These were muscarinic. Okay. And we also said that these were metabotropic, so they were not ionotropic. This is going to be a longer process to bring about a cellular response. Okay, these receptors have to be coupled to a G protein in order to have some cascade of events to bring about this cellular response. It's not as rapid and as quick as this nicotinic receptor here. Okay. Now let's talk about some differences between this synapse here and this synapse here, right? Actually, so this is a little bit different. It's gonna get a little messy here. All right, so what were the differences we said between this synapse and this synapse? This is a neuron-to-neuron synapse, and this is the neural effector junction, right? What were the differences between those two? Right, so we said that the distance was greater, right, in terms of the area that that neurotransmitter had to diffuse across. What else was different? There were presence of varicosity. So there were varicosities along this neuron. And there was not that discrete axon terminal here. Okay. All right. Now just to recap, what was the function of the parasympathetic system overall? That was responsible for what type of body functions? Rest and digest. So I'm hearing it in front here, rest and digest. Right, rest, digest. And sympathetic system was fight or flight, right, or freeze, fight or flight, okay? Now let's talk about the neuromuscular junction here. So we said this was the neuroeffector junction, but this was the neuromuscular junction, where a motor neuron is going to innervate or excite the muscle tissue. So this was a terminal bouton here. So let's have a terminal bouton. And this we said was the motor end plate. Okay, what was the only neurotransmitter of the somatic nervous system? What was the neurotransmitter? There was only one. There's only one neuron and one neurotransmitter. Acetylcholine, thank you. Acetylcholine here. And what was the type of receptor? We said we want a quick, fast depolarization of that membrane. So what type of receptor? Nicotinic, thank you, nicotinic. Okay, so the three places we had nicotinic receptors was on the postganglionic neurons and on skeletal muscle. All right, now we're gonna look more in detail at, let's wrap this up quickly here. The events of, we're gonna look at two things and then we'll wrap it up. And I'll open up for you to ask some questions. We're gonna look at the neuroeffector junction as well as those receptors and their mechanism. Okay. So this is the neuroeffector junction. And this is the neuromuscular junction here. Okay. Okay, this is our varicosity here of a postganglionic neuron. And this is our terminal bouton of our motor neuron. Okay, this is our motor end plate of skeletal muscle. And this is our smooth muscle, cardiac muscle, glans, adipose. All right. Now we said when an action potential gets to this varicosity, there are some vesicles that have the neurotransmitter in them. And again, depending on what neuron it is, this neurotransmitter is going to be different. Okay? If it's a postganglionic on the sympathetic side, the neurotransmitter is norepinephrine. On the parasympathetic side, it's acetylcholine. Now there is a calcium, there's a voltage-gated calcium channel here. When this action potential gets here, calcium rushes into this varicosity, and that's going to cause exocytosis of this neurotransmitter here. And it's going to bind to either a muscarinic receptor or what type of receptor did we say if it was norepinephrine? Adrenergic, right? An adrenergic receptor. Now this can be alpha 1, alpha 2, beta 1, beta 2, beta 3. What did we say about alpha receptors? They were more common. What else did we say about them in terms of their affinity for norepinephrine and epinephrine? They prefer norepinephrine over epinephrine, right? And the beta receptors were different. Beta 1 and beta 3 had equal affinities, but beta 2 preferred epinephrine, okay? Now let's look at the neuromuscular junction side. It's a similar sequence of events here. The only difference is that this neurotransmitter is only acetylcholine, and it's going to bind to a nicotinic receptor here. Okay. Now, just to wrap up really quickly, we also talked about an enzyme, acetylcholinesterase, that was going to break down excess acetylcholine at the junction and return choline back into the varicosity or the terminal bouton, and acetate would float away. Okay. All right, so I'm going to open it up now for any questions. Anything you want me to go over? I didn't get to go over everything I had planned, but I'll definitely give you guys some time to discuss anything in detail. Hmm? Oh, sure, sorry. I'm so sorry. There's one that I drew here. Oh. Any questions on anything? Anything from cells, tissues, integumentary?
I just have a question. The reticular fibers, are they just in the basal lamina or do they extend down to the connective tissue?
The reticular fibers are in the basal lamina. They're a property of the connective tissue. They're connective tissue fibers, but they're going to extend up from the connective tissue layer into the basal lamina. Okay, if we don't have any other questions, I'm going to run through these receptors really quickly for you. Okay, we talked about these 5 adrenergic receptors, right? And we talked about some properties of these receptors. And I'll post this online. I'll post these images online just so you guys can have them. You don't really have to watch the drawing. Okay, so we said alpha 1 and alpha 2 were on some vascular smooth muscle. They preferred norepinephrine over epinephrine, and that alpha 1 specifically had second messengers called IP3 and DAG, and it was a stimulatory G protein that it was coupled to. So it's going to activate these second messengers. right? And as a result, because of the unique location of these alpha-1 receptors, that is going to be an excitatory response to these tissues, right? 5 minutes. So if we think about it as a blood vessel, if you do nothing to it, it'll constrict down on itself, okay? So if you block or inhibit the second messenger, alpha-2, is going to be excitatory to its tissue. So that was alpha-1, which activated its second messenger. Alpha-2 inhibited its second messenger, but both of them were excitatory to their effector tissue. Okay? And they had 2 different second messengers. Alpha-2 had a second messenger of cyclic AMP, but it was coupled to a G inhibitory protein. When we look at the beta receptors, okay, beta-1 was in the heart and lungs, beta-2 was in the respiratory tract and the uterus, beta-3 was on adipose tissue. We said beta-1 and 3 had equal affinities for norepinephrine and epinephrine, whereas beta-2 preferred epinephrine over norepinephrine. Now, all 3 of these receptors have the same second messenger. It's going to be cyclic AMP, and we looked at that cascade of events where it activates adenylate cyclase, increases the production of cyclic AMP, and brings about a cellular response. But only beta-2, because of the unique location of beta-2 receptors, activating that pathway was bringing about an inhibitory response to the tissue. It's going to cause smooth muscle relaxation or smooth muscle dilation. Okay, so the 2 unique receptors that stand out here are beta-2 and alpha-2. And I want you to really drive this point home. Although it is inhibiting its second messenger, that is going to bring about an excitatory response to the tissue. Do not mix the cascade of events at the subcellular level with the effect on the tissue. Okay? Beta-2, again, is going to be a little bit of a tricky one. Although it is activating its second messenger, it's going to bring about relaxation. That's the response. It's still going to be a response there. But in terms of the tissue level, that is an inhibitory response of relaxation or dilation. Okay? And then the others are pretty straightforward. They activated their second messenger and excited their tissues.